Hi Tuesday, Thursday class. Thank you again for being understanding and flexible with these lecture videos. It's definitely, I think, the best way for me to be there in spirit. So today we're going to talk about lipids. And there's a lot of different subcategories of lipids. So if you saw the PDF on Canvas, it's a flowchart of all of the subcategories of lipids and then the subcategories of those. And so really it just gives you kind of a brief overview of the chapter of all of the different labels that we want to be able to use. So first we do want to start by defining what a lipid is. And it's a very broad definition. It's a naturally occurring organic compound that is soluble in nonpolar solvents and insoluble in water. And that insoluble in water is probably the biggest definition that we return back to. And in order for that to happen, the other thing that the majority of our lipids has is a big hydrocarbon component. And so that's what prevents it from being soluble in water. Other than that, though, there's going to be a huge variety of structures and functions. And so that big hydrocarbon component can come from a ring system like the steroids, or it can come in the form of long chains, like down here in this fat molecule. And so we see it in a wide variety of options, which is why there's so many categories and so many subcategories. So we're going to start with fatty acids. And like the name suggests, they are carboxylic acids with a long hydrocarbon chain. So a couple things to take note of. We have a table here with a variety of fatty acids in it. The first column shows us how many carbons they have. Notice the numbers. They are all even numbers. So these fatty acid chains have an even number of carbons. And then when we look at the structures over here, they are all very long carbon chains. The top half are all saturated, so no double bonds. The bottom half are unsaturated, so both can occur, and we can label those as saturated, unsaturated, and then these couple on the bottom here, because they have so many double bonds, we would even classify those as polyunsaturated. The ones with the double bonds are all in the cis conformation, and they all have that one CH2 group in between the double bonds. And then the other thing we want to look at on this chart are the melting points. So within our saturated fatty acids, the larger the structure, the higher the melting point. And this goes back to intermolecular forces, goes back to Chem 1A. When a molecule has that longer chain, it's going to have more surface area, so it's going to have stronger London forces, which means a higher melting point. If we compare the saturated versus the unsaturated, the saturated also have a higher melting point, and it does have to do with stronger London forces. When there's that little kink in the chain because of the double bond, they can't have as much surface area touching their neighbor, and so it's going to have a lower London force, which means a lower melting point. Sorry, trying to advance my slides here and it's not going. There we go. Okay, so our next category are the waxes. And waxes are long hydrocarbon esters. And so they're made from one of our long fatty acid chains and a long alcohol chain. So there's a couple examples down here. Notice that this first one on one side has 25 carbons and the other side has 30 carbons. So these are very large molecules, and this is the major component of beeswax. We find waxes in nature all over the place, and a lot of the times if you think about things that would repel water, it's going to be because they have a waxy surface. So the coating on birds' feathers, the exoskeleton of bugs, the surface area of some leaves and fruit, so the blade of grass here with the beads of water, that is because it has that waxy coating. here is the start of our flowchart. So yes, there's a big giant empty space here that will fill up before we are done. We have lipids at the top, and I know that this font is super tiny, but it's because the chart's going to end up being gigantic by the time we're done. So again, this is on Canvas. You can print it out if you haven't done so already, or look it up online. But at the top here we have lipids, not water soluble, fatty acids, long carboxylic acid, waxes, long ester. So I tried to put a little brief structural explanation for each of the categories. And then of course, feel free to add to that, add more info, draw pictures, whatever's going to help you. Next category are triglycerides. So our triglycerides are made from a glycerol molecule. So it's a 3-carbon chain with 3 alcohols on it. And then these fatty acids. So these fatty acids have come into play now for the 3rd time. And when we combine those together, we make a triester that's held together by this 3-carbon chain. So they are officially called triglycerides. They are also called triacyl glycerols. In my description on my flowchart of what their structure looks like, I labeled them as a triester, just as a reminder of what that structure looks like. These R groups on the end are going to be long hydrocarbon chains. If they are all the same, it's called a simple triglyceride, and if they're all different, it's called a mixed triglyceride. And then our triglycerides have subcategories. We can break these down into fats and oils. Fats are going to be the triglycerides that are saturated or only have one double bond. And because of that, they're going to have strong London forces, so within the molecule, between the chains, and then between 2 different molecules. So that's going to increase the London forces and increase the melting point, which means that it's going to be a solid at room temperature. So this is something like butter. And these items that we would buy are usually going to be animal based. So dairy products that have been solidified. So something like butter or lard. And then the oils, the carbon chains on those are going to be unsaturated chains. So because of the double bonds, they're going to have the little bends in their chains, which means lower London forces, lower melting points, and that means that they're going to be liquids at room temperature. So this is things like olive oil, things that are usually plant-based. So we can turn oils into fats. So another way to think about that in terms of your kitchen is that we can make oils solid. So this is something like margarine. We can take all of those double bonds and hydrogen, palladium, carbon catalyst and turn them into either a saturated chain or just that one double bond. All right, next category are soaps. So if we take that fat molecule, so our fat molecule was the glycerol with the fatty acids attached, and we break it apart, so now we have our glycerol and our carboxylate ion separately, that makes soap. And so we've done this in class. We made soap last semester, I think. And so we still have those long hydrocarbon chains, and we have this polar section of the molecule. So the carbon-oxygen double bond and carbon-oxygen single bond with that negative charge. And then we're definitely going to have some kind of cation, usually sodium or potassium, to balance out that charge. So let's talk about how soap works. Soap usually forms what's called a micelle. So a micelle is a spherical cluster of these soap molecules. And when they form this cluster, they're all going to point in the same direction. So this purple CO2 minus, that is the carboxylate portion of the molecule. And notice that all of those are pointing out. Because they're polar, they will be attracted to the water. And then this little black curvy line here, that's representing the hydrocarbon chain or the non-polar section of the molecule. And all of those are going to point towards the center of our micelle sphere. The orangish blob here, that represents what you're trying to clean. So that represents the ranch dressing that was on your plate. And because the ranch dressing is kind of oily and kind of greasy, it doesn't get rinsed off just by water. So we have to use soap. And so the soap is acting as like a little carrier. It is going to be attracted, or there's going to be an attraction between that non-polar section of the soap and the greasy ranch dressing. And then it's able to wash it off of the plate because the negatively charged portion of the soap is going to be attracted to the water. So it's kind of just like the middleman between the non-polar ranch dressing and the polar water. It can talk to both things. And then the bottom of the slide here has an example of detergent. So detergent is made from a benzene with a sulfone group and a long carbon chain. And so really it's just a synthetic soap. So it's one that can be made or can be manufactured and is not found in nature. And then when this R chain here is a straight chain and not branched, then that would also still allow it to be biodegradable. So we add on to our flow chart here. Here's our triglycerides, which are labeled as triesters, and our fats, which are saturated, and our oils, which are unsaturated. So those are the subcategories under that. And then our soaps are the long carboxylate groups with a cation to balance that out. So our next category of lipids are the phospholipids. And you can kind of see how our structures are developing. So we still have a diester group. So we have that glycerol backbone, and we have these two ester groups, and then we're going to attach to that a phosphate group. And so this is part of the phosphoglycerides subcategory. There's quite a few subcategories for the phospholipids. Of course, there's more than one name. So it's also called a phosphoacylglycerol. Again, it's made from glycerol. It has those acyl groups and it has a phospho group. So it does work. It just happens to have more than one name. So it is going to be very similar to our triglycerides, but that phosphorus group has replaced one of our esters. And there are, again, more than one type down here. These are just examples. You don't need to know these specific ones. It's just showing you that they can look different. Where we see our phospholipids is in cell membranes. And so here is the cell membrane, and just like some of our other molecules, because it has that polar area, so the esters and that phospho group, that is the polar section of the molecule, those are all going to gather together, and then the non-polar chains are going to be pointing down, again, all pointing in the same direction. And then these chains here, if they are saturated, because they can pack closely together, it's going to decrease how much movement there is and decrease the fluidity. And so this can cause things to be packed more closely together. If they are unsaturated chains, then they have more movement, but it also means they're susceptible to breaking down with oxygen. And so this is something that vitamin E can prevent, because vitamin E will react with that oxygen instead. So there's some studies that show that vitamin E can prevent the effects of aging because of this. So it will react with oxygen before our cell membranes do, so it can help your skin look better longer. So that's why you see vitamin E in a lot of lotions, or they recommend using vitamin E oil on scar tissue. All right, another subcategory of the phospholipids are sphingolipids. So these are also found in membranes. The difference here is that they contain an amino alcohol. So we have the amine group and we have an alcohol group. And so this is a small version of a sphingolipid. It's a specific type. This is a bigger version down here, or a bigger example. So we have that double bond alcohol amino portion, and that's what categorizes it as a sphingolipid. So here's the start. We've added phospholipids There's our phosphoglyceride. So they have the two long two long ester chains, excuse me, and then that phosphate group. I have it labeled as PO3X because there's going to be a PO3 group and then something else attached to it. So the X can really represent a lot of different things. And then our sphingolipids had that amino alcohol group. All right, a couple more phospholipid categories. The next one are the prostaglandins. And so the basic prostaglandin shape is that it has a cyclopentane with stuff attached to it. And it's going to have two substituents that are trans to each other. And for the most part, these can be long carbon chains, but there's going to be some variation. These are found everywhere. They're used to regulate physiological responses, so meaning blood pressure and blood clotting. There's a lot of different subcategories, and they're labeled as PG and then some letter, so PG prostaglandin, and then the letter tells us what category they fit into. So here's the ones that we need to know. PGA, B, and C are all enones, so a ketone with that double bond. So if we look at the structures here, that's what they have in common. They have that 5-membered ring. They each have a ketone. They each have a double bond, and then they each have those two substituents because that's part of the prostaglandin definition. And if they aren't on a double bond, then they do have that trans configuration. Over here, because of that double bond, trans just isn't an option. The one underneath here, we have PGD, E1, and E2, and these are all beta hydroxyketones. So again, they all have that 5-membered ring, they all have a ketone, and then in that beta position, a hydroxy group, and then they still have those two R groups as substituents that are trans to each other. And then the last one is PGF2 alpha, and this is a 1,3 diol. So our ring contains the two diols as well as those two trans substituents. All right, here they are on our chart. We have our prostaglandin and then our three, I was going to say subcategories, but I think we're down to a subcategories, but our three types of prostaglandins. Last slide on phospholipids. So I did say there was a lot of them. Sorry, there's a lot of noise in the hallway. I'm sorry if you guys can hear that. All right, so the first category are thromboxans. So this is our thromboxan structure here, and the example is that it contains a cyclic ether. And we see these in our blood cells. They are vasoconstrictors, so meaning that they constrict our blood cells, or blood vessels, excuse me. And then the next one underneath it is prostacyclin, and these are vasodilators, so they make our blood vessels wider. So these two are going to work in tandem with each other. And the prostacyclin's defining characteristic is that it has these two 5-membered fused rings. So again, with both of these, there's a lot of different types, and what these chains are or how long they are is definitely going to vary. But the last bullet point on each of these is what defines those categories. And then the last one in our prostaglandin category are the leukotrienes, and these help contract the muscles in the lungs. So this can be for good reasons, but it can also be because of a heart attack or an asthma attack or even just allergies. And it is a long carbon chain that contains a carboxylic acid and a diol group. So spread out, but a diol group. All right, so there's the end of our phospholipids. We have our thromboxans, our prostacyclin, and our leukotrienes. So we do still have a little bit of white space to fill in here. So the next are the terpenes. And we've talked about terpenes a couple of times because we've used them in lab. And so specifically, I know we've used carvone last semester, and in a couple different labs, you were asked to find the isoprene units. So a terpene again, it's a very diverse group, but what they have in common is that the number of carbons in the structure is a multiple of 5. So 5, 10, 15. You get it. They are found in plants and they get extracted as oils. So the oils that come from, or the oils that are in terpene form are essential oils. So that's where that word originally came from. I don't know if everything labeled as an essential oil still follows that definition or not though. And they are made up of isoprene units. So an isoprene unit is a 5-carbon group, so a 4-carbon chain with a methyl branch at position number 2. If our terpenes contain oxygens, then we can also label them as terpenoids. So a couple of the terpenes down here have been broken up into their individual isoprene units just by their coloring here. So if we look at this first one, here's 1, 2, 3, 4, and then this is our 5th one as a branch. 1, 2, 3, 4, and there's the 5th one as a branch. And so we can see the different groupings. It also doesn't matter where or if there is a double bond, because that is not part of the isoprene definition. So it can have them, it doesn't have to have them, it doesn't really matter. Let's look at the one that has this ketone on it just to see how it affects things. So we have 1, 2, 3, 4, and then the methyl makes the 5th one. So we don't include that in the count because it's not a carbon. And then over here we have 1, 2, 3, 4, and then we have that carbon branch. So same thing as what we've seen in lab. We can classify terpenes based off of the number of carbons that they have. So we have to have 2 isoprene units in order for it to be considered a terpene. So that's the smallest that it can be. So a monoterpene contains 10 carbons or 10 isoprene units. A sesquiterpene contains 15, and this is the category that the essential oils fall into, is when they have 15 carbons or 3 isoprene units. Diterpene, triterpene, tetraterpene, this is based off of the 10, so 20, 30, 40. And then a couple examples here. Squalene is shown down here, and each isoprene unit is highlighted. This is the precursor to cholesterol, so we'll see that in a couple slides here. And then tetraterpene is, or 2 examples of tetraterpene are beta-carotene and lycopene. So these both provide coloring to some natural products. So beta-carotene provides the orange color to carrots and apricots, and lycopene provides the red color in tomatoes and watermelon. And it's really common for us to have molecules that are colored when they have this many double bonds all side by side together like this. And so it will often have a color to it. And that's something we talked about last semester when we looked at the UV Vis data. All right, so there's our terpenes. No little subcategories for it, just our terpenes up there. And we are going to look at how our terpenes are synthesized, and then we'll look at our last lipid category. So when we synthesize the terpenes, well, I shouldn't say we, this is the biosynthesis. So when they're synthesized, we're starting with an acetyl coenzyme A. So we have an enzyme here, and we know that enzymes are really good catalysts, and it's going to actually go through a Claisen reaction to create that first carbon group chain. And then as it's continuing to react, it's going to go through a reduction reaction. So we're removing that coenzyme A from our structure. And then it reacts with ATP. And so this one you've probably learned in biology, but this is adenine triphosphate. And so it has 3 phosphate groups. One of them attaches to that alcohol, and the ATP turns into ADP, so diphosphate. And that happens again, so we gain that an additional phosphate group. And then as the reaction continues, we reduce down our alcohol and our carboxylate group into that basic isoprene unit. So we're going to continue this. So this structure that's down here, this isopentenyl pyrophosphate, is going to be the starting point on our next slide. So we have a couple amino acids helping us out with this as well, and we'll look at amino acids in our next chapter. But we're going to use those to help react with our double bond there and create that carbocation. We're going to do a proton transfer. And so really we've just isomerized that double bond. We've moved it to a new position. This diphosphate group is a good leaving group. So when it leaves, it creates this carbocation. But we're just going to take another one of these molecules, that's what's right here, and it's going to react with that carbocation. And so this is the start of our chain reaction. So it creates this 2-isoprene unit, and that diphosphate group can break off, and this process can continue. So larger terpene molecules can be built as these units continue to be added on. So one of the other things I mentioned was that squalene is a type of isoprene structure, so a type of terpene. It is the starting molecule for cholesterol. So again, this is a biosynthesis, so a nature thing. This is not how we do it in the lab. But squalene starts by reacting with an enzyme, by creating an epoxide on one of those end double bonds. So this end bond here turns into an epoxide. And then if we remember back to when we talked about epoxides, we talked about how to open that ring through basic and acidic conditions. So this is an acidic ring opening. So our oxygen is grabbing onto that hydrogen, and then it creates this huge domino effect. So because this carbon right here is going to lose its bond to that oxygen, we form a new bond here, and that's going to close that ring. This will close that ring, this will close that ring, this will close that ring. So that is the creation of this 4 fused ring system. The base again is going to come in and take off this hydrogen here, which creates a big shift domino effect, but it allows us to have the double bond in place and some of our carbon groups here moved around. This is my favorite part of the mechanism. 19 steps later, magic happens, and we make cholesterol. And so if we look at the difference between these two structures, both of these methyls have been lost, and then the double bond that was right here is shifting to over here. And so it has a very specific structure, but to get this to this takes an additional 19 steps. So this is not a mechanism you need to know. It's really just showing us how these things can happen outside of a lab and actually in nature. All right, our last category of lipids are the steroids. And the defining characteristic of the steroids is that they all contain a 4-ring system, and the rings are fused. And so over here we have the basic system. The rings are labeled as A, B, C, and D. D is a 5-membered ring. A, B, and C are 6-membered rings. Rings B, C, and D are trans to one another, and this is something we talked about quite a while ago, but it was when we first talked about cyclohexane, so maybe back in chapter 6. This also does show us the numbering system, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, so going around A and B, and then 11, 12, 13, 14, 15, 16, 17, so kind of making an S shape around C and D. There are 256 possible stereoisomers of cholesterol because of its number of asymmetric centers, but only one of them exists in nature, and that's because of how specific the enzymes are, those catalysts that help produce it. It's also the most abundant steroid that is found in animals. So that's why it's brought up a lot. So we do have a few subcategories, 5 specifically, of our steroids. So the first ones are the glucocorticoids. And in all honesty, you guys could probably pronounce these better than me because you've taken a lot more biology classes than I have. So try not to laugh too hard at my mispronunciation in this chapter. Okay, so the glucocorticoids are responsible for glucose metabolism. So here is our basic structure of an example. Again, we have that 4-ring system here. And then one way to tell the difference between these 5 is really in comparison. So we'll go through all 5 and then kind of look at some of the differences. Mineral corticoids, they are going to help reabsorb minerals, so sodium and chlorides, by our kidneys. So the name there, the mineral, it literally is describing what its job is going to be. So here's our example over here. And then the next 2 are different types of sex hormones. So androgens are the secondary male sex characteristics. So testosterone's probably the most common one that we hear about from that grouping. And then estrogens are the secondary female sex characteristics. So the example down here is estradiol. And then the last one is progestins, and the most common one out of that is progesterone. And this is a hormone that helps prepare the uterus for pregnancy. And so if we look at all 5 of these compounds, again, they all have that 4-ring system. They all have these 3 hydrogens in the middle that are very specific in their stereochemistry. Our estrogen is the only one with a benzene ring, and it's the only one that's missing this methyl. So I think that the estrogen stands out the most because of that benzene ring. One thing that I think is really interesting is the difference between testosterone and progesterone. The only difference in the molecule is that we have an alcohol versus a ketone. That's the only difference. And that's one of the differences between a male sex hormone and a female sex hormone. And it's just this tiny little difference in the molecule. And then when we go up here and include the differences, so again, our 2 top steroids have that enone, just like our 2 down here do. They have the methyls and the hydrogens. So really the difference is going to come about up here. So our gluco and our minerals both have that alcohol group. This one is the only one that has this aldehyde, and it has this ketone and alcohol. So I think that our mineral corticoids are definitely the busiest in terms of its substituents. And then our glucocorticoids just have that methyl, and then it does have this dual substituent. So the glucocorticoid is the only one with 2 substituents on the same carbon. So however you want to memorize these obviously are going to make good flashcard topics, but there's just some things to notice that might help you recognize these. And there's the rest of our flowchart. So there's our steroids and our different categories down there. I did not put descriptions for these ones because they are a little bit bigger molecules and a little bit more rigid. There's not as much flexibility in what they can contain. So these, like I said, these are going to be really good flashcard topics. How these are going to show up on the test, for the most part, I will show you a structure of something and then ask you what category it falls into. With a couple of them, we did talk about their functions in nature or in our bodies, so those will make good true, false, or multiple choice type questions. I'll let you know more details when I put out the study guide, but these questions on the test are definitely going to be the smaller point questions, so true, false, multiple choice, matching type questions. Okay, that is it for chapter 25. Thank you guys so much, and I will see you on Thursday, and we'll start chapter 21.